second half an hour, grade 12 trace to 24th of June start.
It sounds like you made a documentary like about
It's
now.
like any more
You guys are watching this.
Like, I don't know
It's
I don't know how much longer I have.
Okay, right, going. Okay. So let's presume they hadn't given you the carrier symbol. Okay? They had only given you shaded and not shaded. And you know that it is a sex-linked condition. Then how would you work out what the parents are? Hold on, Charlize. Okay, so it goes like this. Start from the bottom and work your way upwards. All the males have got a Y chromosome. This male shows the condition, X little n. This one doesn't, this one doesn't, that one doesn't. Okay? And that tells you, if you go up to the parents, that must be why this father does not show it, and so he must be X big N. The mother doesn't show it, so she must be X big N. But for that son to have little n, X little n, this mother must be a carrier. Does that make sense? Okay, so you know the males here. The females, you know from that father, must have got X big N. What you're not certain of is from the mother, did they get big X big N or did they get X little n? Because they're not showing you whether they're carriers or not, you have to put both those options. X big N or X little n, because you cannot tell what these are. Does that make sense?
Would they give you like a follow-up question and say one of the daughters was interbred with this
They could.
son, and then say determine whether they X big N, X big N,
Yes.
or X big N, X little N?
Yes. So one of these girls, one of these three females, would have been, oh, you all know these words, Alexandra.
Yeah.
Tsarina Alexandra, who married the Russian Tsar, and then they had the son that had. And therefore, if they had said to you, this was Alexandra, and they had the little boy who was a hemophiliac, so he must have been like that, then they would ask you, they could ask you, what does that tell you about her genotype? So she must have been X little n, X big N, X little n, to have a son like that. So do you see that recessive sex-linked conditions are passed from the mother to the son? The X-linked ones are passed from the mother to the son. So the father would never pass an X-linked condition on to a son, because he always passes the Y chromosome onto his sons. So he's not passing it on. So how would you tell from a pedigree whether the condition you're looking at is a sex-linked condition? Okay, so what you would look at is it being passed from the mother to the son? And is it mainly in males? So generally they will give you a pedigree if they're asking you in a matric exam. It'll only be in males. And that's one point of evidence. But it's got to be a big enough sample. You can't tell if this couple only had like 3 children and one of them's a male and has got it. Then you can't tell. It's got to be a nice big sample like this. Okay.
Ma'am,
Mm.
the female, two from the left, is a carrier, so wouldn't it therefore only be...
No, I said if they didn't give you a symbol for a carrier.
Oh, okay, sorry.
You're awake today. Okay, you had a question. Let's go.
Yes, ma'am, I think close that window. There's a terrible draft coming through.
Okay, do you know that by law I'm supposed to have all the doors and all the windows open? Under COVID regulations. Okay, so I've got one tiny little window, a little bit open. I'm breaking the law. Suck it up. Okay. Right, guys, we're all right. Okay, so from the family tree given above, state the genotypes of Queen Victoria and Prince Albert. We've already done that. Theoretically, what percentage of their sons would be expected to have inherited the trait haemophilia? Show your working. Okay, Charlize, go. Yes, Carmen, go.
I said 25%.
I said 50%.
Because now when I did the calculation, there was XX, 2 X's, and both of them have capital N's.
Okay, no. Tell me the gender of that child.
Also female.
Okay.
There was a male with an X.
So they're
capital N.
asking only males.
Male. Yes, so I had 4 kids, and there was only one male with the X little n y. But then there was another male with X capital
Okay,
N.
it's asking of the males.
Yes. Oh,
So
okay.
ignore the female children.
Okay, then it would be 50%.
Okay, right. You happy, guys? Okay. All right. Do you remember mutations? A little bit. A little bit. Okay. Is everybody okay with pedigrees? Do you need more pedigree problems?
Maybe it's a practice, ma'am. No, it's not.
Okay. So then just keep reminding me. Somebody keeps sending me emails until I've done a worksheet for you.
Yeah. Kia's not here, but she sent an email yesterday about the elephant shoe.
She's not here when I wanted to show you an elephant shoe. That's a fool in me bag. They're all at home in warm belly beds.
Can you show us?
Hmm?
Can you show us what
I don't know, really. I kind of woke up in the morning and then I saw myself in the car. Nah, just takes tomorrow I think I'm in school. Yes, I have something for you. Like a whole day working on the so by 6 p.m.
I feel like I'm more productive than normal.
I've been doing that.
The way I moved during lockdown, I would have never done that at school. Never. She's not your speech, you know what I mean? And it's like sticking in your head. So,
Okay, right. Let's keep going until this thing shows.
that was very easy. But the thing is, I haven't really said That's the thing, but yeah, otherwise, it's much better moving. And usually you can put it on paper. At the end of the day, you know it's in your chapter's done. It's not going to be out of the way to wait for later.
Why don't you talk to Paul and Alice? We can find out. And then we'll find out how on top.
But even for our
Okay, right, let's just quickly have a look while I watch with one eye to see this thing to find where the elephant shoe is. So mutations, remember that mutations are a spontaneous change in the genetic material of an organism. Okay. Who of you guys watched this thing?
What thing?
Wild Earth.
No.
Aren't you so sad?
I used to watch that crocodile. That Australian crocodile.
Oh, no, this is South African. Watch South African. Okay. Oh, stop. Okay. How am I going to do this? It's only on the screen for like a millisecond. Stop putting in front of the camera for it.
But why don't you put it on your big screen?
Because I've got it on my phone and I have to find it.
Why don't you email us?
Because you're going to have to watch 3 hours. Gone.
Again, they would fall prey to snakes, birds of prey, things like tough adders.
Oh, no, I'll have to find it for you. Sorry, Kia. Okay. All right. This is like a real moving thing. Okay, right, let's do this first. Okay, so any change in DNA is a mutation, and it normally occurs during the time that DNA is busy being replicated and when cells are busy dividing. So active tissue, where there's a lot of cell division, more likely to have mutations than cells that are already, that are not actively dividing. So somatic cells that will never divide again. Some mutations can be inherited. Where do those mutations have to be inherited? Ready, steady, go. William?
In the gonads.
In the gonads. Okay. You need to be very specific in the germinal epithelium of the gonads. Okay, because those are the cells that divide and form sperm or ova. Okay?
And did you say in the germinal epithelium?
Yes. Okay. All right. 2 types of mutations. Gene mutations, which means even although they might have like really big effects, they are within one gene, so it's in terms of size, it's much smaller than a chromosomal mutation. So a chromosomal mutation is a huge mutation that affects many, many, many genes. And it can happen, for example, Down syndrome is an example of a chromosomal mutation, and that means a whole extra chromosome in every single cell. So it's a huge mutation. But it can be a chromosomal mutation and a little bit smaller. For example, when two chromosomes separate from each other, two homologous chromosomes separate from each other in metaphase 1, anaphase 1, separating from each other, it's possible for a piece of a chromosome here to stick onto the chromosome that's going the other way. So that means that it's not a whole extra chromosome, but it's an extra part of a chromosome. So it's not quite trisomy, because it's not 3 chromosomes of that pair, but it could be a part of a chromosome extra, sticks on to one of the other chromosomes. Okay, you happy with that? So both of these can occur in somatic cells. Tell me what a somatic cell is. Who are we at? We've done that whole row. We've done that row. Bongo, this is yours.
Try to give an example.
No, because in the matric exam, if they say to you, what is a somatic cell, and you give an example, you don't define it, you don't get the mark.
Well, a somatic cell, or like charity, or
Fortunately, they mark videos in the matric exam. Where you going? It's a body cell.
yeah,
It's not a gamete. It's not a sex cell. Okay. All right. And an example would be a liver cell. Good. Well done. Or germ cells. And germ cells are the same as germinal epithelium. Germ cells. Okay. So germ cells give rise to gametes, or they could even occur in gametes. It is only if a mutation occurs in germ cells or existing gametes that they can be passed on to offspring. So if one of your little skin cells does a little mutation, you're not going to pass it on to your children. Okay. All right. So somatic cell mutations not passed on to the next generation. However, generally organisms accumulate these as they get older, and this is thought to be one of the causes of cancer. But when a mutation occurs in a germ cell or a gamete, then goes on to form a zygote, the mutation is passed on to all the cells of the new individual. Germ cell mutations alter the genome of an organism. And this is how evolution occurred. William?
And so, not related to this, but the previous line, there are multiple genes inside a chromosome, right?
Tell me that wasn't a question.
That was a question.
Makes my eyes very red when I cry, so I hate crying in class, but this is a cause for crying. Okay, look. Okay, remember I showed you yesterday? Look.
Yes.
Remember I showed you just how many gene mutations can occur on the X chromosome?
Yes.
Okay. Are there multiple genes in a chromosome?
Yes.
Good. How many genes do humans have? What have you just answered? What's the question you answered? I swear I'm going to cry. You've answered the question, how many chromosomes does a human have?
Okay.
I asked the question, how many genes does a human have? per cell?
Okay.
And the answer is mhm.
A lot.
Yeah, very many. So, way back in the day, they used to think that humans had about 100,000 or more genes per cell. And then they did what is known as the Human Genome Project, where they sequenced the human's chromosomes. And they were actually very, very surprised to find that a human only has about approximately 20,000 genes per cell. Maybe between 20,000 and 23,000, which is actually not a lot of genes when you consider there's got to be a gene for every enzyme that controls every single chemical reaction in a human body. 20,000 is actually not a lot. So why are you wasting genes on tongue rolling and thumb shape and freckles versus no freckles? It just like seems such a waste when there's so few genes. But anyway.
Now, isn't it like one chromosome, 20,000?
No.
Oh, like
No.
total.
Total on all of those different chromosomes.
Wait, so now if you wanted to work out the genes in a chromosome on average, you would go at 20,000 divided by 46.
Got
So
to
it's
take into
chromosomes
account the length. So because chromosome 1 is much longer than
Right.
chromosome 22,
Yeah.
it's got more genes.
So it's a divides in relation.
Yes. So if you look here, you can actually see that. So chromosome 1 is so long, it's got more genes than chromosome 20, which has got very few genes.
Yeah.
And remember that that is only genetic disorders. It's not actually all of the genes. So it's not showing gene for eye color and that kind of thing. Those are only the genetic disorders that they've listed there. Then the other thing I said to you yesterday is this particular poster was from 1997. And the Human Genome Project, where they worked out all the genes, was finalized in 2003. So this is definitely not a definitive thing. It's just what they knew in 1997. Doesn't mean that they haven't found out more things since then. Okay, right. Is that all the questions at that point? Okay. All right, so blah, 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 blah. Okay. So the effective mutations can be beneficial, okay, or they can be neutral, having no apparent effect at all, or they can be harmful, and it's possible that they can even be lethal. So Queen Victoria's mutation for haemophilia, harmful, and in fact she did have a few children that died. Okay, all right, neutral would be a mutation like everybody used to have brown eyes and then somebody mutated to have blue eyes. Does it really matter? No, it doesn't. It's not a benefit, it's not harmful. And beneficial mutations would be being immune to malaria or COVID. Okay, right, that would be an awesome beneficial mutation. Who had a half a hand at that point?
I don't know.
Go.
I wanted to ask, so Queen Victoria, for example, her mutation did not harm her at all.
Yeah.
So if you were just looking at her, would you not say it's neutral?
No, I think I would say it's harmful because her children got it and died.
So there, when we say beneficial, neutral, or harmful, we're not only referring to the single organism,
Yes.
we're referring to in
Yes.
general. Okay.
That kind of that species. Okay, you guys okay with that? So, blah, blah, blah. Two different kinds of gene mutations. The one you have to study is called a point mutation. And a point mutation is a change in a single base, or very occasionally they will call a point mutation 1 or 2 or 3 bases. So very few changes. It's like only at that point does it change. So remember that many genes code for enzymes or parts of enzymes whose proper functioning relies on their precise shape to be able to work, and even having one amino acid different in a protein may alter the whole shape of that protein and its ability to function. So you've got to know the various types of point mutations. So the ones you know are insertions. Insertions, it's also called an addition, but it's preferred for the IEB for you to call it an insertion. It wouldn't mark you wrong if you said addition, but rather say insertion. A deletion, a substitution, a duplication, and an inversion. Okay, so in an insertion, what happens is an extra nucleotide is added. So if you look here, the fat old cat saw the dog. An insertion would be the, and there's an extra E. Do you see that it changes every, why are my words 3 letters?
Because the what you call is kept in 3 things when it replicates.
Sure. I'm sure everybody knew exactly what you meant. I knew what you meant.
Okay.
Did the rest of the class?
Yeah.
Michelle's shaking her head.
You know the what you call, Michelle? That's it.
Okay, because the codons are in 3s. And remember that for every codon, it would be 1 amino acid added to a protein. Does that make sense, guys? Do you know what I'm talking about? Matthew?
Yes, ma'am.
Okay, so by adding that E there, do you realize that every codon from there on is messed up? Okay, so while these changed codons may code for the same amino acid, it's possible that they won't. Tell me the correct scientific term for the reason why they may code for the same amino acid. It doesn't matter if it was term 1. So when you get to maths and they give you this major problem there, you go, I learned to add in grade 1, Psh, forgot it then. Or English, Shakespeare. I don't read. Sorry, I learned to do that in grade 1. It's over, it's past.
It does muscle memory though. This is especially muscle memory.
No. It's exactly the same type of memory. Go. There's one word for the reason why those codons may still code for the same amino acid. No.
What's the first letter?
Okay. This deletion also has a frame shift. So you can see that the F was lost. The F for fat was lost. And so all of these codons are also wrong. Substitution. Okay, the fat old bat. So C was substituted by B. Let me finish this one and then you can do it. Duplication would be an extra, so the fat, and there was a duplication of the T there. And an inversion here, it was a whole 3 nucleotides